cell and tissue culture. Hello everyone. So today's topic is application of cells and tissue culture. So let me start with that. So the first application is in the form of clonal propagation. See, in nature, the method of propagation, it can be either asexual, right, by multiplication of the vegetative parts, or it can be sexual, right, through the germination or through the generation of the seeds. So multiplication of genetically identical copies of a cultivar by the asexual reproduction is called as a clonal propagation. So I just write the definition over here. Multiplication of genetically identical copies of cultivar by asexual reproduction is called as clonal propagation. So when a population is derived from a single individual by asexual reproduction, it constitutes a clone. So here the concept of clone is also coming. So what is a clone? When a population is derived from a single individual by the asexual reproduction, that will constitute a clone. So we can say that the clonal propagation through tissue culture is popularly known as, we are familiar with this word, that is the micropropagation. So it involves an in vitro propagation of selected genotypes and ultimate establishment of the plants in the field or in the greenhouse. So use of tissue culture for micropropagation, this was initiated by a person, G. Morel, in 1960. So he founded this as the only commercial viable approach for the orchid propagation. So he was working on the orchid propagation. So plants obtained from tissue culture, they are called as microplants. Microplants can be generated in different ways. So these microplants, they can be generated in 3 different ways: from the pre-existing shoot buds or primordial buds, meristem, which are encouraged to grow and they are proliferated. Falling shoot morphogenesis, when the new shoots are induced to form unorganized tissues or directly upon explanted tissues of the mother plant. And third way is through the formation of somatic embryos, which we have already studied in the previous class about somatic embryogenesis. So there are 5 stages which I will be discussing about the micropropagation. Let us do that. Five stages of micropropagation. The first is the preparation of the explant. The second is the formation of the callus. Third is the shoot development. Fourth is the root formation. And the last one is the transfer to the glass house. So if we see a sequence, how it is working, there is an explant, which is like I'm talking about the process of micro propagation. So explant can be the root, it can be a leaves also. So from the explant, there is the initiation of the callus. So here is the callus formation, then embryogenesis of the callus, which will lead to the embryogenic material. So after embryogenic material which is formed, there is a development of the shoots. So there is a shoot development. Then the plantlets are here. They are here without the roots. Then after that, there is a root development. Means the whole plant is formed over here. Then this is transferred to the glass house. So it will harden over here and we get an established plant. So this is the process of micro propagation. This is the first advantage of using tissue culture or a cell culture. The second advantage is the production of virus-free plants. So we know that plants can be infected by a wide variety of microbes, right? So they can be viroids also, they can be viruses, they can be bacteria, they can be fungi, nematodes also. So this infection greatly affect the yield and the quality of the crop or the ornamental plants. In some cases, infection can be eliminated by micropropagation of the unaffected parts of the plant. So the basic method of obtaining a virus-free plant is a culture of the apical meristem. So what is important over here is we need a culture of the apical meristem. So this is important, apical meristem. If the growing tip, because apical meristem is a growing tip, right? So if the growing tip is cultured, there is a possibility of obtaining a virus-free plant, so which can be further multiplied, right? So there are other methods also, like high temperature treatments is given, sometimes incubation of the explant on the suitable culture media containing malachite green or thiouracil, which can reduce the viral infection and replication. So here what I just give you the important point is that apart from using a apical meristem, there are other methods also. For example, the high temperature treatment. High temperature treatment is given to obtain a virus-free plant. Second is the incubation of the explants on suitable culture media which includes or it contains malachite green or thiouracil. So what they will do, they will reduce the virus infection and its replication. So this is the second, you can say, the application of the tissue culture. Third is the plant germplasm conservation. So what is a germplasm? The sum total of all the genetic material which is present in the plant species. So germplasm is the basic ingredient of all the breeding programs. So it provides the raw material, which is the gene, and which the breeder uses to develop a commercial crop variety, right? So there is a conservation. So like this conservation is very important. So plant germplasm may be conserved in the form of pollens, in the form of seeds, or the plant tissue. So we can say that like germplasm is conserved as a seed. It can be stored at an ambient temperature, low temperature, or ultra low temperature. But many crops, they produces short-lived seeds, which are not the best material to conserve. So in addition, like roots, tubers, they also loses their viability rapidly, and their storage requires a large space, right? So here the cryopreservation comes to play. Cryopreservation of the germplasm. This is the most common conservation approach. So what happens over here in this approach, the tissues or the organ may be frozen and stored in the liquid nitrogen at minus 196 degrees Celsius for long-term storage of the germplasm. So this would be a great value in the conservation of the germplasm of those crops which normally do not produce a seeds, because उनके लिए बहुत good है. For example, the roots, the tuber crops, or where it may not be desired stored seeds can be produced. For cryopreservation, the cells are cooled at a very low rate and then they are transferred to the liquid nitrogen for storage, right? So a cryoprotectant is used cryoprotectant like DMSO. Now what is DMSO? It is dimethyl sulfoxide. These are certain examples of the cryoprotectant. So I'm giving you DMSO. Then there is a glycerol, then methanol. They are used to protect the cells from injury due to some freezing, right? So So I hope this is clear. Coming to the fourth advantage or the application, the fourth is the artificial seeds. Artificial or the synthetic seeds, they are defined as an encapsulated somatic embryos that is engineered to be of a practical use in commercial plant production. So synthetic seeds have multiple advantages. Let us discuss about the advantages. So like they are easy to handle. They can potentially be stored for long time. Just a minute. Okay, so they can be potentially be stored for a long period of time, and there is a potential for scale up, low cost of production. This is the third advantage. So they can be stored at 4 degrees for a shorter period, or they can be cryopreserved also in liquid nitrogen for a long term. And the fifth and the last one is the production of secondary metabolites. So we know that plants produces thousands of economically useful chemical molecules such as alkaloids, resins, tannins, and latex. So these chemicals are described as the secondary metabolites. So the function of secondary metabolites in plant is mainly, it is associated with the defense against the pest and the pathogen. However, many of these chemicals, especially the various alkaloids, if I talk about the alkaloids, they are having immense use in the medicines. So such compounds are produced in very small amount, and therefore they are very quite expensive also. So plant cells and tissue culture can hold a very promising role for having a large scale production of these secondary metabolites. So here there is a transgenic hairy root culture that has revolutionized the role of plant tissue culture in production of secondary metabolite. So this is used in production of secondary metabolites. So they are like, these are the unique in their genetic and biosynthetic stability, and that they are faster in growth also and more easily maintained. So usually this methodology, through this methodology, a wide range of chemical compounds have been synthesized, and there is a series of distinct advantage of producing valuable secondary products in the plant through plant culture, cell culture. So, like there is the advantage of using this method is that the production is reliable, it is simple, and it is more predictable. The isolation of the phytochemicals can be rapid and it is very efficient as compared to the extraction from the complex whole plant, right? So interfering compounds that occurs in the field grown plants can be avoided in this cell culture. So another advantage is that tissue and cell culture can yield a source of defined standard phytochemicals in a large volume. So I'll just give you some examples of the metabolites, secondary metabolites. For example, we have the product. So there is a plant source, which is a plant source, and the use. So I'll just share some examples from this. So if I have a product called as berberine, the plant source is Coptis japonica, and they are used as antibacterial. Okay. Another example is dioxins. The plant source is digitalis lenata, and the use is here in the use as a cardiac medicines. So there's other examples also which are mentioned. So I'll just stop over here. So these were some applications of the cells and tissue culture. Hope you are able to get it. Thank you so much.